mysteries. Quote, "The genetic seeds of life swarm throughout the cosmos, and some of these genetic seeds fell to Earth, as well as on other planets. And these genetic seeds contain the instructions for the metamorphosis of all life, including women and men. DNA acts to purposefully modify the environment, which acts on gene selection so as to fulfill specific genetic goals. The dispersal and activation of silent DNA and the replication of life forms that long ago lived on other planets." Rowan Joseph, developer of the evolutionary metamorphosis thesis. What makes humans so special? Perhaps it's because we are the only species to ponder how others perceive us. We have this rational mind and active free will, plus a penchant for self-idolatry. We create architecture, medicine, literature, art, music, and science, yet have the capacity to punish others. We are the only species who can destroy others with the push of a button. How is it that we can be so creatively different above all the animals? After all, our DNA is about 99% identical to chimpanzees. We descended from apes according to evolutionary biologists. Our hands are certainly key genetic traits, allowing humans both precision and power. We can touch all 4 fingers with the thumb. Why do we have such versatile hands? What is it exactly that makes humans human? The genome is a huge field of study and a good place to start to answer these fundamental lifeology questions. Just days after conception, the embryos of chickens, turtles, and humans look remarkably similar. Only as they develop over time do they dramatically change. It's not the number of genes that a life form embodies, but what they can do that makes every species unique. Mutation must also be considered because it is a crucial variation tool for the diversity of life. Since DNA consists of vast quantities of information, it can be rearranged and manipulated to create a whole host of creatures. The DNA is merely a code or a sequence to our genes. The vast bulk of DNA does not process proteins, but regulates the switches which turn on and off genes that create our unique characteristics. DNA proteins create hair, muscle, cartilage, and every other feature in humans. The switches activate at one time and not another, in one place and not another. Other genes give orders to the switches to turn them on or off and when. Timing appears to be everything. Switches form the essential attributes of all species. They tell the cells in the body at what time to activate. What's more, over half of the switches operate in the brain alone. Switches are found in the dark DNA, which is still considered uncharted territory. Sequencing the growth in a fish fin, for example, is similar to the development of limbs and fingers on other creatures. It shows how we can be very similar, yet so different from the animals. Scientists are currently sequencing the DNA of animals and comparing it to other forms of life, including humans. Researchers can reconfigure the switches, sequences, and pathways through mutation to create entirely different life forms. The study of DNA is rising to the challenge of better medicine and a better idea of ourselves, allowing us a clear view of our past and a peek into the future. Monkey to man. To truly understand who we are, Let's begin by identifying human DNA differences with our closest living relative, the chimpanzee. The first noticeable difference in the embryonic stage is that human DNA becomes active early in thumb and big toe development. Brains are the other signature organ which show early differences. Human brains are 3 times larger than chimp brains. Another vital clue as to why chimps are so different is a mutation in the human jawbone, which allows our skulls to grow until we're about 30 years old. Great ape skulls stop growing at 3 or 4 years old. Scientists have identified a gene that regulates brain growth. Compared to chimps, our brains are radically different, with a large series of mutations. For example, there are 15 million letters in our DNA different from chimps. Brain complexity and size is the most fundamental difference. The biggest difference in our mutual DNA are the switches, where in humans more than half are involved with the brain. The chimp to chicken cortex is a 2-letter difference, while there is an 18-letter difference from chimp to human. This relatively small difference, however, has enormous consequences. Human and chimp bodies each have trillions of cells, and each cell has a nucleus of 23 chromosomes. These chromosomes are coiled threads of DNA helix molecules plus protein molecules. Each DNA molecule has a set of nucleotides consisting of phosphate, sugar, base, and nitrogen. And each forms a double-stranded helix where the strands are linked by hydrogen bonds called base pairs between guanine and cytosine, or thymine and adenine. There are 3 billion base pairs in the human genome. Typically, each human being has 100,000 genes. A gene has about 1,000 to several million base pairs. For every gene set, there may be a meme set within humans. A meme set refers to semiotic signs, symbols, meanings, beliefs, instincts, cultural patterns, spiritual patterns, or archetypes. Memes are considered cultural analogs to genes because they self-replicate and mutate. Charles Darwin pondered how there could be so many different species and how some of those species eventually changed to become others. He developed an advanced understanding of life's great diversity. Evolution has been said to be the best idea anyone has ever had. His inspiration came from the Galapagos Islands, where he noticed that the tortoises had different shell shapes on all the different islands. He also noted that finches and other Galapagos bird species were all variations of a single type. Darwin famously recorded 13 species of finches on 13 islands, each slightly adapted for the conditions. Diet patterns of food for each finch determined its characteristics. Genetic variation drives the change for nature. Tiny variations over time and over generations select the best genes, which led to one of Darwin's most famous concepts, the survival of the fittest. Darwin began to look further and wondered if snakes and whales, species once having legs and teeth as seen in embryos, had descended from other animals. All embryos clearly develop in a cephalocudal direction that is occurring in the long axis of the body in the direction whereby the head develops first, followed by the tail or body. Darwin envisioned a tree of life in which all species were related. For example, birds and dinosaurs share many multiple characteristics with each other. as many as they share with their own species. Some say birds are the living legacy of dinosaurs. Darwin's tree of life could be called descent with modification. He saw that dog breeders select traits to create new dogs. Natural selection explains the big diversity in life. Every species is in a desperate struggle for survival. Darwin saw a species battlefield, a pattern for survival depending on their environment, which determined their characteristics. Eventually, monkeys crawled out of trees, began walking upright, and Darwin's descent of man had begun. And while Darwin's evolution concepts largely stand intact today, he did not have the opportunity to examine DNA to take his understanding to a new level. What he would have seen was that DNA can be found in the cells of all species, a veritable code to make all living things grow and develop. DNA does not stay the same in each generation, as half comes from the mother and half comes from the father. Change can also come through mutation, which generates variation, of which Darwin was keenly aware. Scientists can now identify genes that evolved with mutation. They can compare a creature's genes to literally see how evolution works. Humans have 3 billion letters in our DNA. We have 23,000 genes, which is about the same as a chicken, but less than corn. Some plants have more genes than people do. The number of genes in a species is not the central issue, but how they're used. The secret of life. In 1953, James Watson and Francis Crick discovered the double helical structure of the DNA molecule. Unraveling the molecular structure of DNA to identify the double helix was the subject matter of the 1962 Nobel Prize in Physiology or Medicine. Francis Crick, co-discoverer of DNA's double helix structure, discussed at length the pros and cons of an alternative theory on the origin of life, based on the extraterrestrial seeding of Earth with blue-green algae. According to this hypothesis, there was a purpose to the seeding, and it was to ensure the continuation of life. Crick noted that this hypothesis of directed panspermia, proposed, he said, by the physicist Arrhenius at the end of the 19th century, postulates that the seeds of life may have been purposefully spread by an advanced extraterrestrial civilization. Later, after biologists had proposed that an RNA world might be involved in the origin of life, Crick noted that he had been overly pessimistic about the changes of life originating on Earth, even after cracking the DNA code with his astonishing declaration, quote, "We've discovered the secret of life." But he continued to find more credence in the panspermia theory than traditional Darwinian theory, while recognizing its edge of science problem. He said panspermia was at least possible and, more importantly, not totally improbable. The secret is that all living organisms on Earth have the same sort of genetic material in their bodies, called DNA, with the exception of certain small viruses that use ribonucleic acid, or RNA, as their genetic material. Unlike DNA, RNA is almost always a single-stranded molecule, and it has a much shorter chain of nucleotides. RNA contains ribose rather than the deoxyribose found in DNA. RNA is a nucleic acid polymer consisting of nucleotide monomers. DNA is a nucleic acid that contains the genetic instructions used in the development and functioning of all living species within their cells. The DNA segments that carry this genetic information are called genes, but other DNA sequences have structural purposes or are involved in regulating the use of these genetic information. Along with RNA and proteins, DNA is one of the three major macromolecules that are essential for all known forms of life. All DNA is made of 4 amino acids, also called the nucleobases: adenine A, guanine G, cytosine C, and thymine T, simply called the base in genetics. Amino acids are formed into a base pair. It is clear that human DNA can be altered or changed in a myriad of ways. The alterations are called mutations. But while the term mutation typically connotes something negative, some mutations are adaptive and create advances. A number of environmental factors can cause mutations. Current theories state that mutations are the prime cause of evolution. Mutations actually occur fairly frequently and naturally. The purpose of DNA is to provide a blueprint that can be replicated by RNA and then onto proteins. The normal rate of small mistakes in the ongoing replication of a DNA template within the cell happens about once every 100 million base pairs of DNA. But incredibly, human cells have a repair mechanism that recognizes such mistakes. Of more relevance is what geneticists call point mutations, where an amino acid is mistakenly replaced because of an external cause. In the 1980s, the U.S. Human Genome Project was established with the goal of mapping the entire human DNA sequence. It was a daunting project that almost went counter to common understanding. Imagine a very narrow 2-meter long ladder with 3 billion rungs. Each rung is made up of a pair of different amino acids that snap together. There are only 4 possible amino acids that can be used to create each rung of 2 amino acid pairs. The 2 ends of the ladder are then twisted in opposite directions. As it is twisted, the ladder gradually pulls together into a tightly wound, irregularly shaped ball, but the incredibly small area it takes up is infinitesimal. Human brain cells, for example, are about 1/60,000th of an inch in diameter. In the center of each cell, in less than 1/4 of the entire space, is the nucleus of the cell. Virtually all cells in a human body have a nucleus containing a complete 2-meter long DNA strand. Essentially, the mapping of human DNA represents a total of 6 billion amino acids in a linear sequence. By 1998, the job of mapping the DNA sequence was virtually completed, but the identification of the expected and normal amino acid sequences does not mean that we understand the actual function and purpose of each. That is, we know what the amino acids are, but we do not necessarily know their function or purpose. The completion of that research is a long way down the line. Nevertheless, several relevant findings have been made that directly correspond with the idea that cosmic rays, panspermia, and external genetic tinkering have sparked human evolution. DNA manipulation by intelligence. DNA is the most complex molecule known. Human DNA contains two strands wrapped around each other in a helix, and these strands are held in place by the four chemical bases A, G, C, and T. DNA mutation can be caused by radioactivity, including ultraviolet exposure, causing the amino acids to split apart and reconnect, thereby affecting the genes in a chromosome. Scientists have the ability to manipulate DNA by using the following three technologies. 1. Genetic technology and genome study. Scientists can take individual components and map them out. They can track down what the DNA molecules do and then concentrate on genetic manipulation. 2. Computerization. Math algorithms or computer models can be used to design or alter DNA. Researchers can create cause and effect models representing scientific optimization. And number 3, nanotechnology. This is the ability to manipulate DNA on an atomic level. Scientists can alter, change, or modify DNA on a molecular level. It is therefore theoretically possible to control or rewrite DNA, but researchers would have to get at the source code. Biological evolution operates at a snail's pace compared to technological evolution. Technological evolution is called the Lamarckian inheritance model, which can be implemented within a single generation. Biological evolution is Darwinian and requires many generations of differential reproductive success, and sometimes takes thousands of years for a single mutation to enter the general population of a species. But let's not forget that the cosmos is very big and very old, with virtually an infinite amount of time in which to operate. Genetic engineering, recombinant DNA technology, gene splicing, DNA modification, or genetic manipulation, GM, are terms applied to the direct manipulation of a living organism's genes. Genetic engineering is not to be confused with traditional breeding techniques where the species genes are manipulated indirectly. Genetic engineering uses the laboratory techniques of molecular cloning and transformation. Genetic engineering endeavors have had some success in improving crop technology, the manufacture of synthetic human insulin through the use of modified bacteria, the manufacture of erythropoietin in Chinese hamster ovary cells, and the production of new types of experimental mice, such as the onocomb mouse or cancer mouse, for research purposes. However, what is really astonishing is that a group of researchers working at a human genome project believe that 97% of non-coding sequences in human DNA is no less than the genetic code of an extraterrestrial life form. Cloning is usually done by taking a nucleus out of one cell of a living being and then transferring it to an egg cell from which the nucleus has been removed. Most of the cloning research has been achieved behind closed doors, and the results are not being disclosed. Cloned people could already be walking among us, but the public is genuinely appalled by a technology that plays God with human cells and sperm. It is possible now to clone your cat before it dies by removing a bit of tissue, taking out the nucleus from one cell, and putting that nucleus into the egg of a female cat from which the nucleus has been removed. The egg would then be implanted into a surrogate cat that would carry the fertilized egg that has the genetic constitution of your own cat. Well, if all goes well, and it often does not, the cat would produce a kitten genetically identical to your original cat, enabling you, in principle, to have the same pet forever. This procedure is not guaranteed, though, as some mitochondrial DNA of your pet would not be transferred in this procedure, and somatic cells in the body undergo mutations during the life of an organism. Therefore, the skin cell of your cat would not be genetically identical to the fertilized egg that produced it. Furthermore, any farmer will tell you that the more you clone the same species, the weaker the DNA gets, and subsequent offspring will be weaker and prone to more diseases. There is also the question of whether the same spirit would attach to the newly cloned being. A challenge to evolution. To better understand the immense block of geologic time, that is, the eras, periods, stages, epochs, ages, and the long march of all conceivable historic time, let's consider a model. Planet Earth's age is generally put at 4.6 billion years, which is the age of the oldest discovered meteorites. To better appreciate the vastness of geologic time, let's think of those 4.6 billion years as represented by a single year. The oldest fossil records go back only about 40 days out of that 365-day year. Indeed, more than 88% of Earth's history is Precambrian. Of the remaining time when the Earth has been home to animal life as we know it, the part that includes proto-humans and their ancestors is less than 1%. The human presence on the planet is only about 2 hours. The presence on the planet of Homo sapiens sapiens, which are modern humans, is only about the last 6 minutes. According to an article in Nature released in June 2017, findings in an old mine at Jebel Irhoun in Morocco date the earliest Homo sapiens appearing around 300,000 years ago. They were definitely around elsewhere in Africa 260 to 160,000 years ago. At this time, Homo sapiens coexisted with Homo erectus in Asia and Neanderthals in Europe and the Middle East. Nuclear atom is the most recently known common ancestor of today's modern human, having lived sometime between 90,000 to 60,000 years ago. Using the one-year model, it makes the notion of evolution possible, probable, and comprehensible for the development of older animal species, but not so much for quickly evolved modern humans. For the scientific community, intelligent design represents creationism's latest grasp at scientific legitimacy. Accordingly, intelligent design is viewed as an ill-conceived attempt to corral science to within religious ideology. But we can now see that intelligent design can be formulated into a scientific theory with empirical support and devoid of religious commitments. Evolution theory based on Darwin's Origin of the Species is in the process of being disproven based on the thought that the DNA molecule is far too complicated to have been formed simply by natural mutations alone. Later, even Francis Crick proved mathematically that it would seem impossible for evolution to produce human DNA in the brief amount of time allowed. The way evolution works on a specific species is on a mass scale and in a much longer time frame than is now afforded, especially for modern humans. Ascension, a leap in potential, that is, the sudden creation of a new being, is not only restricted to the Christian concept of rapture, where Jesus will return and usher in 1,000 years of peace. It could be a cosmic event that simply prompts another stage in evolution, as such occurrences have been prophesied in almost every major religious and spiritual tradition on earth. Such could be a time when our DNA will multiply into new strains, and humans will naturally redevelop dormant superhuman abilities. What the mind can conceive, the body will achieve. ET genes in human DNA. The non-coding sequences of DNA are a common attribute of all living organisms on Earth, from algae to fish to people. Non-coding sequences, initially known as junk DNA, were discovered years ago, and their complete function still remains a mystery. In human DNA, they constitute a much larger part of the total genome, says Professor Sam Chang, the Human Genome Project Group leader. According to the study group, the overwhelming majority of human DNA was off-world in origin. They found that apparent extraterrestrial junk genes could just enjoy the ride with the hardworking active genes that are passed from generation to generation. After comprehensive analysis and with the assistance of other scientists, computer programmers, mathematicians, and other scholars, Professor Chang wondered if the apparently junk human DNA was created by some kind of extraterrestrial programmer. The alien chunks within human DNA, Professor Chang further noted, have its own veins, arteries, and its own immune system that vigorously resists all our anti-cancer drugs. Professor Chang further commented that, quote, if we think about it in our human terms, the apparent extraterrestrial programmers were most probably working on one big code consisting of several projects, and the project should have produced various life forms for various planets. They also have been trying various solutions. They wrote the big code, executed it, did not like some of the functions, changed them or added new ones, executed again, made more improvements, tried again and again, end quote. Professor Chang further stipulated that, quote, our hypothesis is that a higher extraterrestrial life form was engaged in creating new life and planting it on various planets. Earth is just one of them. Perhaps after programming, our creators grow us in the same way we grow bacteria, in petri dishes. We can't know their motives, whether it was a scientific experiment, a way of preparing new planets for colonization, or a long-term ongoing business of seeding life in the universe, end quote. The team of researchers working with Professor Chang furthermore concluded that the apparent extraterrestrial programmers may have been ordered to cut all their idealistic plans for the future when they concentrated on the Earth project to meet the pressing deadline. Very likely, in an apparent rush, the extraterrestrial programmers may have cut down drastically on a big code and delivered a basic program intended for Earth. Professor Chang is one of a growing number of DNA scientists and other researchers who have postulated an extraterrestrial origin to humanity, who said, sooner or later, we have to come to grips with the unbelievable notion that every life on Earth carries genetic code for his extraterrestrial cousin, and that evolution is not what we think it is. Professor Chang further concludes that when we see in our DNA is a program consisting of two versions, a big code and a basic code. He bravely affirms that first fact is the complete program was positively not written on Earth. That is now a verified fact. The second fact is that genes by themselves are not enough to explain evolution. There must be something more in the game. Star Child Skull. The Star Child skull is a genuine 900-year-old bone discovered in a Mexican cave in the 1930s. It sat in a closet for many decades before it was examined by experts and put to the rigors of DNA testing. Ongoing research has provided proof that the Star Child skull possesses physical characteristics, biochemical attributes, fibers, and a residue inside the bone that indicate it is not human. More recently, a 2010 DNA test result confirms that the skull is at least partially of extraterrestrial origin. Researchers have concluded that the skull presents at least 10 standard deviations from the norm. It is comprised of bone uniformly half as thick and weighing half as much as normal human bone, but is significantly more durable. The bone is more of a substance like tooth enamel. A DNA test in 2003 found that it has human matrilineal lineage, which confirmed that it cannot be a pure alien species. However, the picture that emerges from the latest round of DNA testing is that the skull once belonged to a human-alien hybrid. The MT is the part of DNA passed only through the maternal line. In 2011, the geneticists working on the Star Child skull discovered that its mtDNA was radically different from human DNA. The maximum number of mtDNA differences between all humans is 120. The Star Child skull has between 800 and 1,000 differences. This is a partial result, but it is definitive enough to declare that the skull's mtDNA is not human. The greatest benefit of discussing the Star Child enigma is to open our perspective beyond the evolutionist or creationist dogma. The stakes are too high to discount this remarkable specimen as a human deformity, rather than as a possible missing link in the human-alien hybrid scheme of things. The issue becomes more of a case of admitting that it is only language that restricts our understanding. For example, considering the statement, "We do not come from apes," we now know that we did not directly descend from apes, although perhaps in some general aspect we did, since we have some shared DNA factors. The emerging perspective is that Earth was populated with creatures in a sort of genetic safari park, overseen by numerous ET genetic tinkering groups. The DNA results of a hybrid ET existing on Earth nearly 1,000 years ago, along with very anomalous features in our human blood and our human brain, all combine in the new intervention theory, gaining more credibility every year. DNA from outer space. DNA building blocks actually created in space was a NASA report headline in August 2011. If asteroids are behaving like chemical factories, cranking out prebiotic material, you would expect them to produce many variants of nucleobases, not just the biological ones. due to the wide variety of ingredients and conditions in each asteroid, said Michael Callahan at NASA Goddard Space Flight Center. Goddard and his team ground up samples of 12 carbon-rich meteorites, 9 of which were recovered from Antarctica. The team found adenine and guanine, which are base components of DNA called nucleobases. Also, in 2 of the meteorites, the team discovered trace amounts of 3 molecules related to nucleobases. The 3 molecules were purine, 2,6-diaminopyrin, and 6,8-diaminopyrin, the latter 2 almost never found in earthbound biology. The discovery adds to a growing body of evidence that chemistry inside asteroids and comets are capable of making building blocks of essential biological molecules. Astrobiologists are fully aware that many viruses are manufactured in the stars. Indeed, the medieval Latin word for influentia signified influence of the stars. In January 2010, another study announced that 8% of human genetic material comes from a virus. This news suggests that human evolution is much more likely to be influenced or even driven by cosmic factors, which is consistent with what scientists have known for some time, namely that viruses are manufactured in interstellar space. Researchers at the Goddard Space Flight Center discovered portions of DNA on chunks of crashed space rock in both Antarctica and Australia. The molecular extraterrestrial visitors contained various types of nucleobases, which are thought to be essential in the creation of DNA and life in general. The scientists were able to isolate the compounds and prove that they did not originate on Earth. The team also found that certain space rocks, depending on their makeup and speed, worked like manufacturing facilities for these biological precursors. The implications of the discovery are far-reaching and suggest that the origin of life on Earth may owe its existence to a well-placed meteorite, and that without it, the planet might still be a rocky, watery wasteland. NASA's new discovery about the elements of life originally being brought to Earth by incoming objects confirms what Swiss UFO contactee Billy Meyer published in 1988. Meyer scooped NASA on this story by a few decades, explaining at the time that the insemination process from comets and meteors by which life started on Earth is a phenomenon that occurs throughout the universe. Indeed, the news reports about NASA's discovery virtually repeats what Meyer said decades ago about the universe-wide phenomenon of life-seeding objects. What these findings suggest is that DNA is a product of a quantum energy wave and is written into the basic laws of the universe. These are the laws that govern the formation of life on Earth, but also govern the behavior of matter and energy in the cosmos. Thus, DNA begins as an energy wave and does not require any physical molecules to exist. The structure of DNA, therefore, exists throughout the universe. We are all of a cosmic and galactic design, not a product of random mutations on Earth. It can logically be deduced that humans have independently evolved in many different worlds. We know there are at least 22 separate types of DNA within the human species. This would allow a genetic memory of other entities who are part of us through many generations of interbreeding. The human design would seemingly be intrinsic to the Milky Way galaxy and probably a fair amount of the entire universe. Our solar system exists on the outer fringe of the Milky Way galaxy. It could then be presumed that other humans have advanced spiritually and technologically much farther than we have here on Earth. If this is the case, then those humans could have colonized Earth in the times before or during the existence of Lemuria or Atlantis. And their skulls reveal great brain capacities significantly larger than ours. As we will learn, these people were largely wiped out by a self-inflicted cataclysm caused by nuclear war between rival colonies. The survivors built pyramids to heal and stabilize the Earth on its axis in the aftermath of this catastrophe. They had direct knowledge of a natural cycle that propels each inhabited planet through quantum evolutionary leaps. They were aware of the physical pineal gland in the human brain that governs ESP and can be activated by this natural cycle. The masters already know this. For ages, esoteric and spiritual teachers have known that our minds, in which the body and its organs virtually reside, not the other way around, are programmable by language, words, and thought, from which instructions to the body then follow. Our minds being extensions of the infinite one mind then explains our seemingly human unlimitedness, even on the level of the material world. In another case in which esotericists got it right, light codes have now been scientifically explained. Light codes are energy patterns, sacred geometries, colors, sound waves, light patterns, and frequencies that can all emit creation. DNA is affected or activated by light codes, which can be received in a myriad of ways. Once you have triggered Kundalini energy, the process continues for the rest of your life. That is because it begins to shift your DNA into alignment with your higher or more authentic self. Of course, the frequency has to be precisely correct, and this is a reason why everyone is seemingly not created equal and can't replicate identical results with the same strength. The individual person must work on their inner processes and their own maturity in order to establish conscious communication with their DNA. It could be that the metaphysical claims that light codes are being delivered to Earth to repair DNA may not be so far-fetched. At the moment, the logic is that viruses only cause DNA mutations, and since there are various interruptions to our DNA, intervening sequences or blanks, introns, repressor molecules may attach to promoter regions. Who is to say that light codes are not inducers that activate genes by combining with repressor molecules as part of the delivery of evolutionary energies that can be used to upgrade our DNA in readiness for the next evolutionary leap? Another intriguing frequencies theory is their influences on weather. Our weather is strongly influenced by the Earth's resonance frequencies, the so-called Schumann frequencies. These same frequencies are also produced in our brains, and when many people synchronize their thinking, or when a powerful individual like a spiritual master can focus his thoughts in a laser-like fashion, then scientifically speaking, it is not at all surprising that they can influence the weather and other events. If the cosmos can deliver agents that cause DNA mutations, why do we not consider that the cosmos periodically deliver genetic material that reverses the decline of the human genome? Within certain parts of the metaphysical community, it has been accepted as common knowledge that light codes are now being delivered to this planet, but the implications have not been well understood. This research could totally shatter the Darwinian model of random evolution in favor of an energetically driven model. This model has nothing to do with creationism and is not religious in nature. The higher an individual's consciousness is developed, the less need there is for any type of device, such as light frequency. One can achieve these results by deep connection to one's inner self, as proven by spiritual masters throughout time. But it doesn't end there. Russian scientists also discovered that our DNA can create invisible, structured patterns in the vacuum energy of space, thus producing magnetized wormholes. These wormholes are the microscopic equivalents of the so-called Einstein-Rosen bridges in the vicinity of black holes, originally observed as being left by burned out stars. The DNA attracts these bits of information and passes them on to our consciousness. This process of hypercommunication is most effective in a state of relaxation. Stress, worries, or a hyperactive intellect prevents successful hypercommunication, rendering the information distorted and useless. In fact, one can simply use words and sentences of human language to influence DNA. This too has been experimentally proven. Thus, living DNA substances present in living tissue, not in vitro, will always react to language modulated laser rays and even to radio waves if the proper frequencies are being used. Brain DNA changes through life. By charting the human brain's genetic activity from before birth to old age, studies reveal that the brain continually remodels itself in predictable ways throughout life. In addition to uncovering details about how the brain grows and ages, these results may help scientists better understand what goes awry in brain disorders such as schizophrenia, Alzheimer's, ADD, and autism. Researchers of a study released in October 2011 focused not on DNA, but on when, where, and for how long each gene is turned on over the course of a person's life. To do this, researchers measured levels of mRNA, a molecule whose appearance marks one of the first steps in executing the orders contained in a gene, in post-mortem samples of donated brains that ranged in age from weeks after conception to old age. Interestingly, the DNA remains virtually identical in each cell's raw genetic material, yet different patterns of mRNA levels distinguish the brain from a heart, for instance, and a person from a mouse, says Ninad Sestan of Yale University School of Medicine and co-author of a leading study. Essentially, we carry the same genes as mice, he says. However, in us, these genes are up to something quite different. To see what those genes have encoded, the Yale study examined mRNA levels of different genes in 57 brain samples. The team divided the brain tissue up by region, so they were all able to get an idea of genes' behavior in different parts of the brain. A parallel study headed by Joel Kleinman of the National Institute of Mental Health in Bethesda looked at gene behavior in 269 brain samples from a single region called the prefrontal cortex. Both studies found a large amount of variation in gene behavior at different life stages, but the prenatal period stood out, demonstrating massive changes in gene activity. Prenatal genes pumped out big quantities of mRNA, production that abruptly slowed after birth, and with development slowing over time. Curiously, many of these genes that slowed down immediately after birth show a surge of activity as a person gets older. The biggest changes naturally occur in the fetus, and then they go virtually unchanged until midlife. But then, when a person is in their 50s to 70s, expression changes pick up again and become quite dramatic. What's more, the difference in gene behavior between male and female brains were greatest at the early stages of development. Some of the genes found to be busier in male brains have been linked to schizophrenia, autism, and other disorders that are known to be more prevalent among males, the researchers noted. These disease-associated genes are very active early on in development and less so as a person ages, suggesting that within these conditions, something goes awry very early. Although gene behavior is incredibly dynamic, the results suggest that brains are more alike than different. Despite millions of differences in DNA, brains have a very common biochemical shape, according to Joel Kleinman. Two people who have very different DNA makeup don't necessarily have different gene behavior in the brain. These individual genetic variations, they do matter, no question, he says, but overall, genes behave almost identically from person to person. That's a phenomenal realization. It means that we're much more alike than we are different. Another study was performed by Dr. John Hawks, an anthropologist from the University of Wisconsin, who has studied and analyzed the last 5,000 years of mummies and grave sites that he has excavated. His research concluded that our DNA has changed structurally by an astonishing 7% in the last 5,000 years. That means that human evolution has already rapidly sped up on a genetic, measurable level. Scientists can trace it in the DNA. Just in the last 100 years, there has also been something called the Flynn effect, which is that IQ scores have had to be continually readjusted every decade because people keep getting smarter and smarter. Junk DNA. Only 10% of our DNA is being used in the process of building proteins. It is this subset of DNA that is of greatest interest to Western researchers and is being examined and categorized. The other 90% is considered junk DNA, the so-called dark matter of the genome. DNA switches are not genes, but they turn genes on and off. Key body-changing genes come from the dark DNA, but evolution leaves behind tiny remnants, the turned-off codes that are no longer in use. This explains how manatees, whales, and snakes could have evolved away their legs. Some genes are known to rule over other genes. They regulate what to do and when. We are told fish are the ancestors of all four-legged creatures and every bipedal human. But how could a fish develop legs and walk on land? Fin-like arms, or arm-like fins, were first used to drag a prehistoric creature to safety on land. Similar genes in other animals trigger growth of arms, legs, and fins. Lungfish, for example, use their fins to drag themselves onto land. DNA switches would trigger the development of longer arms and legs. DNA is now less mysterious than it has been in the past, and many geneticists think that so-called junk DNA regulates the expression of other genes. Yet, we know that large portions of DNA are viruses that infected non-replicating portions of the genome and thus became trapped in the host's genome. Some scientists suggest that the human genome is nothing but a vast cemetery of viruses. Much of the junk DNA is constructed of replication viruses, basically genetic messages which say nothing more than copy me. But where do the viruses come from? Scientists have focused on only 10% of our DNA, those that are designed to build proteins, thus leaving a large gaping hole in the interpretation of the other 90%. A few Russian researchers convinced that nature is intelligent joined linguists and geneticists in a venture to explore the remaining 90% of the junk DNA. Their results, findings, and conclusions are simply revolutionary. These scientists can prove that DNA can be reprogrammed by words and frequencies similar to light codes. According to their findings, our DNA is not only responsible for the construction of our body, but also serves as a communication system and for data storage, similar to a biological internet. The Russian linguists found that the genetic code, especially in the apparently useless 90% of DNA, follows the same rules as human languages. To this end, they compared the rules of syntax, which is the way in which words are put together to form phrases and sentences, the study of meaning in languages forms called semantics, and their basic rules of grammar. They found that the alkaline of our DNA follows a regular grammar and has a set of rules, just like our languages. So human languages did not appear coincidentally, but instead as a reflection of our inherent DNA. Author Elaine Smitha raises some interesting questions about junk DNA in her book. If you make the rules, how come you're not boss? She inquires, perhaps junk DNA contains all the secrets of the universe, including those unconscious potentials you have yet to discover. Perhaps it is this DNA that allows you to travel without your body into starry realms. Maybe this is where you go in your dreams. Quite possibly, intuition, psychic powers, and remote viewing, non-local information gathering, fall into the realm of this amazing communication channel. Perhaps with a relaxed mind, you can tap into the prime DNA alphabet soup for transport. It's absolutely fascinating to consider. Ghost DNA. Author David Wilcock describes a living energy field, source field, that forms what he calls the fundamental building block of all space, time, energy, matter, and biological life. According to Wilcock, such a field may be the crystallization of a united symbiotic consciousness available to us via our DNA. The source field, as it is directly related to biology, includes all life, even the simplest of life forms, such as and even including space dust. The concept that DNA is formed in the process of planetary evolution and space dust may sound absurd, but it can be proven by something known as the DNA phantom effect. Dr. Vladimir Pupinin put DNA in a tube and shone a laser through it. To his surprise, he realized that it captured the light and caused it to spiral through the helix as if it were a crystal. Even more amazingly, when Pupinen removed the DNA, the light continued to spiral on its own. Thus, he could conjecture that there is a specific distance from the sun where the tiniest spiraling waves will gather a planet's natural materials together to form the DNA molecules of life. The DNA is the wave, and the wave is the DNA. For light to form a DNA helix on its own would seem impossible. That is, unless the light itself had become harmonically tuned to some naturally existing frequency in the energy of space. This suggests that the spiraling light energy of DNA was there first, and the physical molecules simply formed around the spiraling energy once the planet was at the proper frequency position. Thus, it could appear that the galaxy is tuned to create DNA. Life itself is an emergent phenomenon of quantum physics. In 2011, Nobel Prize winner Duc Montagnier proposed that our DNA can electromagnetically teleport itself. Montagnier described a phenomenon in which DNA emits electromagnetic symbols of its own construction called ghost DNA that can be mistaken by enzymes as the real deal and replicated in another place. Essentially, it is DNA teleportation. To demonstrate this, Dr. Montagnier used two vessels of sterilized water, one with a small amount of DNA, one without. He then periodically electrified a 7 hertz electromagnetic charge to both vessels. After 18 hours of zapping the second vessel, it then contained all the signatures of the DNA. Hydrogen and oxygen molecules in sterilized water became DNA. Another study by Professor Ignacio Pacheco mixed sterilized bleached sand with regular water in a test tube and turned up the heat to 1,000 degrees. He sealed and sterilized the sample again with distilled water. After 24 hours, a little scum was seen growing on the surface of the glass. Without sterilizing of the sand, the experiment would not have worked. So, where did the DNA come from? DNA appears to be created or written into the source field. The source field is the source of all life because it does not appear to be randomized life as Darwin proposed. DNA is a quantum emergent phenomenon. As Russian scientific research directly or indirectly explains, along with decades of replicated experiments at the Institute of Noetic Sciences, phenomenon such as clairvoyant intuition, spontaneous and remote acts of healing, self-healing, the effects of affirmation techniques, the existence of unusual light or auras around certain people, especially spiritual masters, and even the mind's influence on weather patterns, and much more. Patanjali in the Yoga Sutras describes these expanded human abilities as siddhas, as simple side effects of the practice of regular, disciplined, deep meditation. This could be thought of as meditation penetrating deep into a person's life essence, or DNA. In addition, there is evidence for a new type of medicine, as discussed by Bruce Lipton in Biology of Belief, in which DNA can be influenced and reprogrammed by words and frequencies without cutting out and replacing single genes. The Russian biophysicist and molecular biologist Pyotr Garjavev and his colleagues also explored the vibration behavior of DNA. they reported living chromosomes function just like solitonic holographic computers using the endogenous DNA laser radiation. This means that they managed, for example, to modulate certain laser ray frequency patterns to influence DNA frequency and thus the genetic information itself. Since the basic structure of DNA alkaline pairs and language are of the same structure, no DNA decoding was necessary. Within the concepts of three-dimensional and coordinate time, a universe is creating itself, and that universe exists concurrently with our own three-dimensional and coordinate space realm. It is not a parallel reality or tucked away in some far corner of the universe. It is right here, right now, only shifted out of phase with our spatial reality so that our physical senses do not detect it. However, our non-physical senses can detect it and operate within it, and this gives rise to some people's psychic abilities. For example, precognition is nothing more than seeing something in the distance, in the temporal landscape. Telepathy is two people standing next to each other in time chatting, regardless of how far apart they are in space. Telekinesis is just a manipulation of the temporal component of an object within your temporal arm and watching how time changes space. Clairvoyance is as if using a pair of temporal binoculars. Virtually all of the extrasensory abilities are easily understood once you realize that you exist in two different realms, a spatial, material one for the body, and a temporal, cosmic one for the soul. And the interesting aspect is that these aren't extrasensory, magical, or metaphysical abilities, just a natural consequence of biological life that we can either choose to learn, use, and develop, or simply ignore. DNA has also been found to have a bizarre ability to put itself together, even at a distance, when, according to accepted science, this should not happen. Yet, scientists are reporting evidence that, contrary to our current beliefs about what is possible, intact double-stranded DNA has the amazing ability to recognize similarities in other DNA strands from a distance. Somehow they are able to identify one another, and the tiny bits of genetic material tend to congregate with DNA having similar characteristics. The recognition of similar sequences in DNA chemical subunits occurs in a way not understood by science. There is no known reason why DNA is able to combine the way it does, and from a current theoretical standpoint, this feat should be chemically impossible. This recognition effect may help increase the accuracy and efficiency of the homologous recombination of genes, a process responsible for DNA repair, evolution, and genetic diversity. The new findings may also shed light on ways to avoid recombination errors, which are factors in cancer, aging, and other health issues. As of yet, no explanation has been offered, although these attributes of DNA lead some researchers to the conclusion that our DNA is, in effect, conscious. Superhuman DNA. In the not too distant future, if not already, scientists will discover the manipulated gene from ancient times in the DNA chain responsible for rapid aging in human beings. Preparations in this area began in 1994. However, whether scientists make their discovery public and utilize their findings to its fullest potential is questionable. In a world already grossly overpopulated, having people's lifespans triple or quadruple would compound the already strained resources when providing for over 7 billion people. It is virtually certain that a new DNA information code will be discovered in the human body, and that the first concrete steps will be undertaken to eliminate afflictions to the elderly, heart diseases, and general physical attrition. A genetic reverse manipulation process will release the human population from the premature aging curse. Genetic manipulations are reversed by retro manipulation of the pertinent gene. Conflicts will also arise within human populations because of their seemingly immortal lifespans. That is, the increase in human longevity will amount to potential lifespans from 150 to 200 years, or possibly even longer. This increase in longevity will also precipitate greater problems of overpopulation and other subsequent obstacles that will include migrations and new types of interbred humans. In spiritual terms, from our current 2 strands, there are 12 postulated strands of human DNA that extend into the 12 dimensions of reality. Beyond the changes to DNA in a laboratory of what is postulated, humans are observed to be naturally evolving to include these new strands of DNA. In April 2011, it was revealed that British toddler Alfie Clamp became the first known example of a human with 3 strands of DNA. Such a finding would suggest that the human race is currently mutating to add an entirely new strand of DNA. It is speculated that humans long ago contained 12 strands of activated DNA, but 10 became deactivated and turned off. These inactive 10 strands are thought by some to be our junk DNA. Similar to what is said about our brains, only a small percentage of our DNA is being used, while the rest apparently lies dormant. It is suggested that humans have 3% of our DNA active. With more activated DNA, we should be able to understand far more, at least from a consciousness point of view. For example, the 12 strands of DNA correspond to the 12 extended chakras in the human body. Future generations with new strands of DNA could even potentially change from carbon-based beings to crystal-based entities. DNA antennas. The DNA antenna in our cell's energy production centers, called mitochondria, assumes the shape of what is called a supercoil. This supercoil of DNA looks like a series of Mobius loops. These Mobius supercoil of DNA are hypothetically able to generate scalar waves. Most cells in the body contain thousands of Mobius supercoils generating scalar waves throughout the cells and throughout the body. The term scalar was coined by Nikola Tesla at the beginning of the last century as part of a powerful non-Hertzian energy that is without frequencies, which he referred to as cosmic waves. Einstein gave reference to the scalar energies in the 1920s. Since then, Tesla and other independent researchers have come to understand both the physical and theoretical model of a scalar wave. They concluded that it is a dipole that is of 2 opposites unified as a toroidal vortex. It spirals in motion and travels in circles. It simultaneously attracts and repels. It resonates and causes to resonate. It is found throughout the natural world. Thus, it is a galaxy, it is a hurricane, it is a tornado, it is a seashell, it is the torus, and it is also DNA. Scalar waves are produced when two electromagnetic waves of the same frequency are exactly out of phase. That is, they are opposite to each other, and the amplitudes subtract and cancel each other out. Vortex in, vortex out. The result is not exactly an annihilation of magnetic fields, but a transformation of energy back into a scalar wave. The scalar is created when two common electromagnetic waves come together from two different converging vectors or angles. When the energy vectors meet, the equal frequencies cancel each other out, leaving a standing or stationary energy. The space the scalar occupies is not a vacuum, but alive with checked and balanced energies. It can be created by electromagnetic generators or naturally when similar frequency waves in the environment meet from two different vectors. Therefore, small random scalar energies are always present in the environment. Scalar waves propagate through the dimension of time, but not through the dimension of space. They are observed to carry information and have a fractal-like structure. Microbiology experts tell us that upwards of 97% of our human DNA is apparently unused, but some are seeing that more and more of the human DNA chain is unzipping for any who are capable of handling the new fourth density energy structure. These people may not physically look any different, but internally they will develop greater depths of emotion, perception, intuition, sensitivity, and what can be termed superhuman abilities. As mentioned earlier, it now appears that DNA can be influenced and reprogrammed by words and frequencies. Frankenfoods. The amazing discoveries about DNA also have a dark side, in the form of creating new DNA and, by extension, new life forms. A biological patent is a patent relating to an invention or discovery in biology. The 1970s marked the first time scientists patented methods on biotechnological inventions using recombinant DNA. It wasn't until 1980 that patents for whole-scale living organisms were permitted. In Diamond versus Chakrabarti, the Supreme Court overturned a previous precedent allowing the patentability of living matter. The subject for this particular case was a bacterium that was specifically modified to help clean up and degrade oil spills. In recent decades, genetic scientists have discovered a protein sequence specified by a segment of DNA that can be modified. New versions of a protein, for example, can be produced by changing the DNA sequence of a gene. The companies that make these discoveries then own the modified genome and are able to patent these new sequences. In the case of basic crops, the companies are gaining control of foodstuffs, controlling food production on a large scale, and reducing agrobiodiversity to a few varieties. The only apparent interest in promoting this technology appears to be economic, despite the claims of seed companies such as Monsanto and Novartis, who say they seek to solve the world's food shortage. A closer examination of this issue shows that the problem is not the lack of food on a worldwide scale, but its distribution, aggravated by prohibitive tariffs by rich nations. Genetically modified crops cannot reduce hunger. The majority of genetically modified crops are destined for animal feed to meet the higher demand for meat consumption in developed countries. There is no evidence that genetic modifications have ever served the needs of humankind, despite all the promises from the companies who stand to profit. However, Even with regard to this technology's great potential, many have raised concern about the introduction of genetically engineered plants and animals into the environment and the potential dangers of human consumption of GMO foods. Critics point out that these organisms have the potential to spread their modified genes into native populations, thereby disrupting natural ecosystems, as has already happened. Immune responses, photons, and torsion waves. We live in a dangerous world. Pathogenic microorganisms threaten us continuously, the 2011 Nobel Medicine panel said, describing the work of scientists over the decades and their understanding of our natural defenses. American Bruce Buechler and French biologist Jules Hoffman, who studied the first stages of immune responses, share the $1.5 million award with Canadian-born Ralph Steinman, whose discovery of dendritic cells in the 1970s was fundamental to understanding the body's next line of defense against disease. The immune system exists primarily to protect living organisms against infection, but it can also protect against some cancers by targeting rogue cells before they proliferate. The first line of defense, innate immunity, can destroy invading microorganisms and trigger inflammation. If microorganisms break through this defensive line, adaptive immunity is called into action. It produces antibodies and killer cells that can destroy infected cells. These two defensive lines provide good protection against infections, but they also pose a bigger risk, and inflammatory disease may result. As when the immune system goes into overdrive and attacks healthy tissue, leading to autoimmune inflammatory diseases such as type 1 diabetes, multiple sclerosis, and rheumatoid arthritis. The effect is often compared to friendly fire, as when military troops hit their own comrades in combat. A variety of information suggests that our DNA's primary function is to store photons. There are about 1,000 photons per DNA molecule. When there is an area of a patient's body that is sick, it loses photons. As a result, the patient's DNA no longer contains photons and their condition worsens. Studying photons in the DNA is how doctors will be able to measure where sickness resides in the body. After all, every physical body consists of elements. Ordinary matter is made up of atoms, and these are made up of protons, neutrons, and electrons. A chemical element consists of those atoms with a specific number of protons in the nucleus. This number is called the atomic number, as is featured on the periodic chart of elements. Modern science has revealed that the atom, previously regarded as the smallest particle, can actually be split. Ordinary matter consists of atoms bound together by an electromagnetic force to form molecules. These molecules come together to form solids, liquids, and gases. A photon is a basic unit of light and is considered energy. An electron travels near the speed of light. When a photon is absorbed by the atom gains the photon's energy. The electron uses this energy to jump up to a higher orbit. The photon energy equals the electron energy in a larger orbit, less the electron energy in a smaller orbit. Thus, if a photon strikes an atom, the atom can absorb the photon and its energy only if the photon's energy is exactly equal to the difference between two orbital energies. This enables the jump from the smaller orbit up to the larger orbit, called a quantum jump.
Finally, it should be pointed out that all forms and shapes and patterns in the material universe are made up of torsion waves. Torsion waves flow in and out of all physical matter, and atoms are torsion wave generators. The counter-rotating phi spiraling electromagnetic or EM waves in the implosion physics that spiral into the nucleus of the atom likewise cancel the electromagnetic components of the EM waves and results in a torsion wave. Torsion waves are energy waves composed of nested phi spirals of light, that is, electromagnetic radiation. Yet they have the capacity to travel far in excess of the speed of light. But actually, they don't travel at all, because in reality, there is no distance traversed. It is analogous to a string on a guitar that strums. There is no actual distance traveled linearly, but the strings vibrate in amplitude waves. Are crop circles made by energy transfer via torsion waves pressing down on the crops? Do DNA or cells communicate via torsion waves? Is communication the medium for telepathy via torsion waves? Can torsion waves also travel faster than the speed of light, arriving almost instantaneously? Now that we have reached the essence of who and what we really are, it's time to switch gears and examine the rich tapestry of human experience on Earth. To begin with another examination of the expansive subject matter in this lifeology section, we need to start at the beginning, at the moment when people first began to conceive a vision of themselves.